Dean, do you send out a link later?
Yeah, to the recording, Lee. Yeah, we do.
Okay, great.
Yep.
It looks like people are still coming on, so I'd wait a few more minutes.
Yeah, no, definitely. Sounds good.
It's pretty good for an August talk here.
Yeah, you are
Yes.
battling
Yeah,
last
yeah,
week of vacation for everybody.
exactly. Yeah, right before
I feel like school starts so early though for a lot of families these days. I remember growing up, it was always after Labor Day, and now like kids are back in school already. So
No kidding. It's really bizarre.
well, good news. The Department of Education is being canceled, so maybe we won't have
Yeah,
school.
that is some good news.
That's good news.
Luckily, they've given us fall weather,
Oh,
so we
yes,
can all
exactly. It'll be endless summer with climate change.
exactly.
I can go away. They don't.
I better turn off my audio just to keep me from going off. Being triggered.
People are still pretty actively coming in.
Yeah, we can definitely give it a couple minutes. This is great.
Yeah, I think we can call it 12:05 and then probably just go from there.
All right.
Perfect.
Where did you grow up, Audrey?
in the suburbs of LA. So
Okay. So in LA, it started after Labor Day?
when I was growing up, yeah, but I actually don't know now what it's like.
Yeah. I know my niece lives in Florida, and I think they go back like mid-August.
Yeah, here too in Sac. Yeah.
Wow.
Yeah, I grew up in Florida, and I feel like the start date was always like August 20th, like right around there.
Really? That sounds horrible to me.
We felt the same, Mickey. We felt the same.
Yeah.
Would it be okay if I just tested my slides real quick?
I'll go for it. Absolutely.
Absolutely, yeah.
How does, oh, continue to share. How does that look?
They look
Perfect.
Great. Perfect.
Okay, so yeah, it's 12:05. So we can get started. Dean will also do the recording. But I just want to also introduce myself for those who don't know me. So I'm the new research program coordinator here at the ADRC at BU. I'm very excited to have our first talk featuring Dr. Audrey Fan. So I'll tell you a little bit about her and then we'll get into the talk. So Dr. Fan is an assistant professor of biomedical engineering and neurology at UC Davis, and she has her PhD in electrical engineering and computer science from MIT, was a postdoc fellow and instructor at Stanford. She's a recipient of numerous different fundings and grants, and she's currently leading the imaging core for the Alzheimer's Disease Research Center at UC Davis Health. And her overall goal is to really bridge engineers in her lab with clinicians and scientists so that we can advance just neuroimaging technology overall and promote brain vascular health across your whole lifespan. So without further ado, I'll let Dr. Fan take the stage with her talk called the Non-invasive MRI to image cerebral perfusion and blood-brain barrier function in aging and dementia. So then we'll do the talk and then we'll have about 15 minutes after for a Q&A.
Great. Thank you for the kind introduction, Samantha. And I'm very delighted to be here today. Thanks for carving out time in the summer. And feel free to interrupt. I might pause at certain points in the presentation just to ask for questions to hopefully make it more interactive. Samantha already covered a lot of this, but I currently am at UC Davis. Our lab is in the city of Davis, near the main campus and near one of our research 3 Tesla MRI scanners. In Sacramento, which, I mean, depending on traffic, is 15 to 30 minutes away, we have our health campus. And I'm part of the neurology department through which I lead the neuroimaging core for our ADRC. And because Samantha mentioned to me, this colloquium was targeted to rec scholars as part of the audience, I just wanted to throw in a little bit of a small personal history. So I am an engineer by training. I spent a lot of time in Boston and really miss it, especially in the summer and the fall, doing my PhD at MIT in electrical engineering. And then I did have the great opportunity to return back to California in the Department of Radiology, where I worked with a clinician, a neuroradiologist. We were not studying aging at the time, but I think that understanding of intracranial stenosis and how to image with PET MRI was really seminal in getting me interested in some of the more physiological questions you'll hear about today. And I just want to point out, I think I owe a lot to the training fundings. So I applied for a K-99 and eventually got it, not on the first try. I did several rounds of faculty search, also didn't get it on the first try. And I just wanted to put that out there for any younger postdoc scholars or rec trainees. Persistence, especially in this funding climate, is really important. And even before, it was really tough. So hang in there. And I started at UC Davis in January 2020, right before COVID hit. So it was a little bit of a remote and I would say a little bit lonely start, but I'm very happy to have my colleagues in the ADRC now to actually meet physically and have a community. My son Paul is featured here. He's almost 3 now, and he was born in 2022. That's a little bit about me, and if anyone wants to chat about any of these components of my background, happy to do so after. But today, in terms of research, I wanted to really focus on imaging of the vascular brain. And you may or may not know that the human brain is uniquely metabolically hungry. So it depends on continuous oxygen and nutrients through cerebral blood flow. It's only 2 to 5% of our body mass, but it takes about 20 to 25% of our cardiac output. And so we may know that in stroke and other more acute events, right, this can have devastating consequences. And it's really important to understand how this blood flow and also oxygenation, the counterpart, so how oxygen is extracted by the brain tissues together form the hemodynamic normal function for tissue viability in the brain. So this sort of methodology was something I worked on, for instance, during my PhD. In the context of aging, as I just mentioned, there are lots of devastating consequences of stroke, affecting over 800, nearly 800,000 people in the US each year. And even if you are able, thankfully, to survive a stroke, 75% go on to have cognitive impairment. So this is a clear causal link in terms of vascular damage and eventual cognitive decline. And there's a lot of longitudinal studies that are coming out also studying midlife vascular risk factors such as hypertension and diabetes, and that tends to correlate with eventual cognitive impairment as well. So one facet of understanding dementia and Alzheimer's disease is kind of understanding that it's not just a clear picture of the disease pathophysiology, that it can coincide with vascular changes and vascular contributions to dementia, and that these may or may not intersect in terms of the mechanistic pathways. I think that's still being teased apart at the moment. The current structural imaging that's non-invasive and able to detect vascular disease in the context of aging and dementia has its limitations. And you may have heard of white matter hyperintensities. So for instance, on a flare, for instance, T2 inversion recovery MRI, these are unexpectedly bright spots that tend to occur in the white matter, and at these key locations, for instance, near the ventricles. These are structural markers, and it's difficult to understand what's underlying these structural lesions. So we think it's potentially vascular because there are associations with certain vascular risk factors, for instance, that have been shown between imaging and other clinical correlates. However, there are questions about topology. So certain areas of lesions may associate with, for instance, amyloid beta accumulation more so than others. So is it really just vascular? And these tend to occur quite prevalently in aging, with over 90% of older adults in advanced age showing these hyperintensities, even if they don't have cognitive impairment. So I think the impact on cognition is also heterogeneous and hard to understand just from looking at and identifying the lesions itself, because it's, again, hard to understand what it actually is happening to create these lesions. So even in this context of a structural vascular lesion that is well characterized, it's important to ask the question, well, can we understand what's happening better in terms of lesion evolution by directly imaging physiology and some function associated with the brain? And could that happen even earlier than the lesion formation? So today I want to talk about one approach using MRI to look at this sort of physiology. And I'll talk about markers of perfusion and blood-brain barrier, specifically revolving around arterial spin labeling MRI and how it can be used more broadly for long-term impact. So the first topic study that I want to chat about is associating cerebral blood flow and progression of white matter hyperintensities, which has been done in a cohort from our ADR. Yes. Is there a question? Maybe not. As background, as we age and also in dementia processes, there are changes in morphology of the microstructural level. For instance, at arteriosclerosis, changes in capillary tortuosity and density that affect how the tissue can ultimately be perfused, including perfusion, how it responds, so supravascular reactivity and blood-brain barrier. And we think this process ultimately manifests in regional changes. So blue here is a schematic from this review paper that indicates changes in Alzheimer's disease, which show patterns of hypoperfusion or reduced perfusion as a result of some of these morphological changes in the brain vasculature. The method that we've been using and many other groups have used is called arterial spin labeling MRI. And what's great about it is that you don't need to inject any tracer. MRI does not have ionizing radiation. Instead, ASL uses arterial blood water in our bodies itself as an endogenous contrast. Very generally, how it works is that RF pulses are applied at a labeling plane below the imaging slice in the brain where we're interested. And so these spins are so-called magnetically inverted or magnetically tagged. And we wait a user-defined post-label delay. So that's a time that allows us to wait for the labeled spins to travel into the imaging slice and perfuse into the tissue. And then we take another image called a control image without the labeling. And the difference of control minus label results in a map like this. So these are axial slices of cerebral blood flow. You can see gray-white contrast here because the gray matter has about 2.5 times the perfusion of white matter. And each voxel, the signal intensity is reflective of the perfusion within that voxel. But overall, it's a challenging technique, although there's been a lot of readout and labeling advances to make it more robust. In general, it's a challenging problem because only 5 to 6% of each brain tissue voxel is blood volume. So you can imagine that it's quite a small signal, but we have advanced readout strategies to be able to make perfusion maps like this. And ASL has been also extended to a multi-delay strategy. So I mentioned you have to wait a post-label delay for the tag to get into the brain and ultimately perfuse into the tissue. But sometimes it's difficult to know what that time should be because a person with an intracranial stenosis or even in aging may have longer arterial transit times due to the tortuosity of the vessels, for instance, that we just talked about. And so we may not capture exactly the right timing, so to speak, to get a perfusion map. Instead, if you're able to collect multiple post-label delays, then you can fit a so-called kinetic model, and it will still give you a cerebral blood flow map, but it has then been corrected for the arterial transit time, the arrival into the tissue, exchange from the blood compartment to the tissue, and then decay. So taking into account each of these dynamics with multiple post-label delayed snapshots helps us get even more reliable perfusion images from ASL. In the literature, ASL has been used in larger studies such as the Human Connectome Project Aging to look at how perfusion, for instance, in the gray and white matter change across age in healthy aging. So for instance, this is over 200 participants with typical aging, a span of about 60 years in the population. And both in terms of this regression and the quartiles of age that are depicted here, you see that CBF, cerebral blood flow, has been inversely associated with age, so a decline with age, especially in the gray matter. And ASL has also been used in studies to understand differences across the AD continuum. So these are results from a recent paper by the UPenn group, where you can see differences in cerebral blood flow on a group level. If you compare, for instance, cognitively unimpaired groups, amyloid negative versus positive, or cognitively unimpaired amyloid negative with MCI or dementia, right? And you see this regional pattern that is very striking. And in fact, areas like the precuneus and PCC, it kind of looks quite like the default mode network for any of researchers who are studying fMRI. It's very interesting that these sort of regional patterns can also show up along the AD continuum. So before I move into our study, are there any questions? Just take a quick pause.
And everyone can feel free to, I was going to say, just unmute or put in the chat, whatever.
Yeah, I guess I just have a brief kind of general question. So one of the studies that we're working on here is the Clarity study. So of course, like being in charge of neuroimaging, I was just wondering kind of your thoughts like on the study, how it's going for you all at UC Davis, and if you guys are a part of this study and kind of how that's looking.
Yeah, I will briefly mention Clarity at the end too, but I think Clarity is really great in that it harmonizes and has as part of, I mean, not the required protocol, but as part of the ADME4 protocol, right? The multi-delay ASL. So I think what's great there is the PET, right? Clarity is often, touted for amyloid and tau PET and the fact that it has longitudinal follow-up. And I think it'd be fantastic to kind of look at the PET markers in tandem with ASL across groups like kind of here, but also on a longitudinal basis. So I am also very excited about Clarity here.
Wonderful, wonderful, us too.
Right. So in our study, we recruited 300 participants. This is a retrospective study from our ADRC. And what's great is that in this cohort, 90 of the participants actually returned for a second MRI where we assessed white matter hyperintensities about 2 years on average after their baseline. And this was recently published by Ice Tamasar. She was a visiting scholar actually with our ADRC visiting from Thailand. What we did is we took our baseline MRI data, we performed segmentation of lesions and actually identified individual lesion clusters as well as dilated in the white matter, so constrained to white matter tissue, penumbra around white matter hyperintensity lesions. And these are areas, regions of interest that we defined based on spatial distribution, although we can talk about, maybe a more physiological or microstructural way to define penumbra. But the idea is penumbra is tissue that doesn't look like a lesion yet, but it's spatially next to the lesion. And so it may be uniquely at risk or vulnerable. There may be early pathophysiological processes happening there, even though clinically you don't see a lesion yet. And we also looked at different locations of these lesion clusters, such as juxtaventricular, like right next to the ventricles, periventricular, and then also in the deeper white matter. What we saw first when we analyzed ASL results in these different regions is that we saw a graded decrease in cerebral blood flow, particularly from the lesion having the lowest relative cerebral blood flow compared to normal appearing white matter, and that even in the penumbra, so whether it's 4 millimeters or 8 millimeters around the lesion, there were deficits, significant reductions in cerebral blood flow in a graded fashion. And this is synergistic with a hypothesis that Dr. Maillard in our ADRC has proposed over 10 years ago, where the core injury, the lesion, may be where, there's actually a flare lesion present and, the worst white matter integrity, so to speak, but that the penumbra is also vulnerable, even though it doesn't show up as a lesion quite yet. So it's consistent with a continuous view of vascular injury, especially with pathophysiology that's not isolated to lesions. And we associate the cerebral blood flow measures with log transformed volume of these white matter hyperintensities and saw an inverse relationship. So, this is all at baseline, but a larger lesion may be representative of more severe vascular injury, or perhaps a lesion that's been there longer because of its size and having reduced cerebral blood flow, particularly in periventricular areas in the green. So these are all at baseline, and this inverse association is suggestive of, a relationship between progression and white matter hyperintensity blood flow. But we wanted to test that directly because we know that at follow-up, different lesions may have different properties in terms of growing, stagnant, or having an incident, like a new lesion form that was not observed on the baseline scan. So we wanted to then leverage the 90 participants who had follow-up and try to understand how does baseline cerebral blood flow relate to this progression structurally of the lesions, whether they grew or not. And these were our major findings. It's a lot to take in, but I want to highlight that in primarily periventricular and juxtaventricular areas, so closer to the ventricles, the CSF spaces, that growing lesions had a significantly lower cerebral blood flow at baseline compared to stagnant lesions. And this was observed not only in the lesion itself, but also in the penumbra. Oops. And then for new lesions, I think this was more challenging to detect because new lesions just tend to be smaller. But also, perhaps in the periventricular space, we're starting to see some effect of lower baseline cerebral blood flow being predictive of new lesions forming. So this, I think, was very exciting to us. We did try to look at cerebral blood flow measures on the 2 time points itself, but that was not significant. And I think it has to do with, 2 years being not a long follow-up, and especially for a physiological marker that we're trying to still understand a lot of the characteristics of it. So blood flow longitudinally, I think, is challenging to track because it can vary. Like, for instance, if you had coffee this morning, your blood flow reduces by 20 to 30%, right? So going back to Dean's question about follow-up, I think there's still questions about the stability and also the timing at which physiological changes occur. But at least at baseline, we were able to see baseline physiology was predictive of how the lesions responded in terms of growth. So to summarize, cross-sectionally, we showed different tissue types that had different blood flow patterns, so including white matter hyperintensities and penumbra in a graded fashion had lower blood flow compared to normal appearing white matter and gray matter. And longitudinally, that baseline perfusion in the lesions as well as the penumbra was predictive of whether they would grow or expand, indicating a likely cause for their development. And as we continue looking at blood flow measures in lesions, we're trying to adopt multiple delay ASL to be more rigorous in the cerebral blood flow measures and see if that, for instance, helps with longitudinal follow-up. And it gives us a separate measure of arterial transit time, like what the transit to the lesion tissue looks like. And ultimately, we are starting to associate the lesion blood flow and penumbra blood flow with cognitive scores as well. This is a snapshot from an AAIC poster that a researcher from our group just presented this past summer, but it's still early days. What's interesting is even in the normal cognitively normal group indicated by blue here, we're seeing perhaps that an higher cerebral blood flow in the lesion areas corresponds to higher episodic memory, maybe indicating a protective effect or, a reduction leading to poor cognitive performance. So I'll take a pause there. This is the midpoint after completing the study. Are there any questions about this study?
Hi, this is Ning from BUMR Core. I have some technical question. So you focused on the white matter hyperintensity, and assuming you do multi-delay ASL. So I'm wondering if you consider T1 of the tissue.
Yeah, that's a really good question. So in this initial study, we actually used single delay ASL. The more recent poster we presented at AAIC is starting to look at multiple delay ASL. But it's challenging because I think, as you probably know, being an MR expert, the T1 of the lesion is longer, right? So it appears dark on the T1 weighted image. And the T1 decay of the tissue is really important as well. In terms of modeling, what we have tried to do is take different assumptions for T1 of tissue and see how it affects these measures. And actually, to me, it wasn't initially clear whether changing the T1 tissue would actually alter the arterial transit time that is estimated. But what we're seeing is actually the blood flow measures are not as affected and in the lesions, whereas arterial transit time is by different assumptions of T1 tissue. So I think it is something that you have to pay attention to, although I think the limitation is we don't have a direct measure in our MR protocol of T1 tissue or just T1 mapping itself that is reliable. So I'm hesitant to just, adopt a brand new value in these regions without really measuring it. But I think that would be a valuable study. And I think it will also depend on how you actually measure these values. So I think we're using FSL Basel as our tool for fitting with multi-delay, but I think certain protocols you might fit ATT first and then kind of do the full kinetic model. And so I think there's a consideration of like what model you actually use. Does that answer your question, Ning?
Yeah, I'm just curious whether you consider T1 because it's affect a lot in the modeling. So yeah,
Yeah.
thanks.
Yeah. And what we've observed in a few cases so far with multi-delay is like actually the ATT surprisingly was affected, which I didn't expect necessarily, like that the estimate of the timing of arrival would change more so than the overall perfusion, but worth investigating more. Yeah, it's a great question.
Thanks.
Cool. Okay, so on to the second half of the talk, where I want to describe ASL as another avenue to look at a different biomarker, which is water exchange across the blood-brain barrier. And many of us have seen diagrams like this about, the unique blood-brain barrier that is protective in the CNS. It's this multicellular interface between blood vessels and brain tissue that includes astrocyte and feeds endothelial cells that are separated by tight junctions. And overall, it plays an important role to regulate influx, efflux of nutrients, proteins like amyloid beta, waste products, and also immune cells. And so because of this critical role, it can be involved in many processes like protein passage into the brain, perfusion, neuroinflammation, all of these that have been implicated in Alzheimer's disease and dementia. For understanding the BBB, there are biochemical approaches, so biofluid approaches to assess globally what blood-brain barrier function is like in individuals. And that includes, for instance, plasma albumin. But while these are more accessible, some of these are very indirect, may require invasive sampling of, like, for instance, CSF, and they're regionally non-specific because you get, one global measure from the neurofluids. Whereas neuroimaging has the opportunity to be more direct in assessing permeability across the blood-brain barrier. It is costly, but I think it has the value of giving you regional insight into blood-brain barrier function. And for schematic purposes, I wanted to just touch on gadolinium, for instance, which is a contrast agent used in DCE, dynamic contrast enhanced MRI. And the idea is that in aging and in dementia, that loss of structural integrity of the blood-brain barrier, and in particular, like tight junctions, can lead to crossing and leakage of gadolinium, these larger particles across the blood-brain barrier, affecting the DCE signal dynamically. Whereas in ASL, where we actually look at water molecules are regulated by aquaporin 4 across the blood-brain barrier. And in a dysregulated blood-brain barrier, it could be both structural changes, but also earlier, before changes in BBB structure, if there is lower function or lower expression of aquaporin 4, that can also affect the passage of water across the blood-brain barrier. So it's a smaller molecule. It has different mechanisms through which it can pass across the blood-brain barrier, which is why we think it's an interesting approach on top of what's perhaps more known contrast-based methods. So these contrast-based methods, I would consider a traditional method. I don't know that it's necessarily a gold standard, but it's one that has existed for a while. It does involve dynamic T1 imaging with injection and dynamic imaging, T1 weighted for both like an input function, like here from the sagittal sinus, as well as in the tissue. And if you can deconvolve this relationship between the input function and the tissue curve, you get maps across the brain of blood-brain barrier function, specifically K-trans, which reflects the ability of gadolinium to the transfer rate across blood-brain barrier. And my colleague at the UC San Diego ADRC, Emily Reyes, recently published a couple of papers, one in Alzheimer's and dementia, where she showed that using DCE, there is BBB breakdown even in cognitively normal participants, older participants, if they are, for instance, at risk, so ApoE 4 positive, they might have higher presence of higher K-trans levels, and that this correlates with changes you can understand from the microstructural imaging with DTI, for instance. So DCE is an established method. Maybe less known is how arterial spin labeling can actually give insight into the BBB. So I mentioned that this is not tracking gadolinium now. It doesn't require a contrast agent. Now we're actually trying to track the exchange rate of endogenous water across the blood-brain barrier. And these two spaces in the two tissue compartment models, the vascular space and the tissue or extravascular space, will have different properties like T1. And Ning just pointed out the T1 also varies in different lesions and tissue types, and diffusion properties, etc. So these two compartments of blood versus tissue are important to understand. And you can use these different properties to separate the compartments. So for instance, USC has developed an ASL-based method where you do ASL, but you also have a diffusion prep module with two different B values, so that when you're playing the higher B value, higher diffusion weighting, you essentially crush out the signal from the vascular space and you maintain the signal from the tissue space, and that allows you to separate the different compartments. There's also the DEBI consortium, which is initiated primarily from Europe. And this is a multi-echo ASL acquisition. So it still uses ASL, the control tag that we talked about earlier, but it incorporates a T2 prep multi-echo acquisition so that you can leverage T2 differences and T2 like multi-dimensional fitting, again, to separate two compartments. And the goal is that once you're able to separate blood and tissue, which together contribute to this overall blue signal that we looked at before with the multi-delay compartment model, now we're separating it into the blood compartment and the tissue compartment dynamics. And that will actually allow us to measure a parameter called KW. It's water exchange across the blood-brain barrier, across these two compartments. The interesting thing is that these techniques are really new, and in recent years, they've been separately used to try to understand how BBB KW, the water exchange across the blood-brain barrier, may change with age. And so, for instance, with the diffusion-weighted ASL, you're actually seeing a decrease in water exchange in this large age range and almost 200 participants of cognitively normal individuals, especially in the parietal lobe. They're seeing this decrease in KW with age, which the study interpreted as, for instance, reduced aquaporin 4 expression with age. However, separate studies that use multi-echo ASL, again in about 200 participants, showed a different effect. So actually the y-axis here is time of exchange, which is the inverse of KW. So actually this is indicating reduced water permeability, and this is indicating increased water permeability with age. I would say the multi-echo ASL is perhaps more consistent with DCE measures, but DCE again is not a gold standard and it's measuring something different. And it's a little puzzling that these two ASL methods, although they're both purportedly measuring water exchange with different physical mechanisms, they have shown in the literature these opposite effects. So our recent study, I'll fully admit, does not solve this controversy, but we wanted to ask the question, okay, if we do these two ASL measures in the same individuals, what do we see? And so we recruited a small cohort. This is our pilot study of young participants and then older participants who are cognitively normal from our ADRC. This is my PhD student who actually recently defended 2 weeks ago. So this is a labor of love from his PhD. And what we did is we directly compared diffusion-weighted ASL and multi-echo ASL in the same people. And what we saw in the same people was consistent with the discrepancy in the literature. So with diffusion-weighted ASL, we saw a decrease in KW in the older participants compared to younger. And we saw the opposite effect in multi-echo ASL with the same people. And we saw this in multiple lobes as depicted here. You will also notice that the range of KW, again, despite it being technically the same biomarker, the range is different and the spread is different across individuals too, which is something we have to look closer into in terms of harmonizing some of the imaging across these two methods. When we looked directly comparing at these two markers, these two ASL measures. So I forgot to mention, so when you separate the blood and tissue compartments with these more advanced ASL approaches, you can measure water exchange, KW, but you still get the original measures we were interested in, like blood flow and arterial transit time. And the puzzling thing is, especially with cerebral blood flow, there's a positive relationship between these two. So ATT is measured slightly differently between the two protocols, but still between ATT and cerebral blood flow, they're showing consistent trends, especially, for instance, a reduction in cerebral blood flow in the elderly group compared to the younger group. But when you directly correlate KW, which is the blood-brain barrier water exchange measure, they are inversely related, which is, again, not originally expected. These are what some of the cortical maps look like. So there's diffusion-weighted ASL where you see, again, the reduction, and it looks quite diffuse in terms of reduction in KW across many brain lobes. And here's, again, the multi-echo where you actually see an increase in KW, so more leaky to water, endogenous labeled water. And here you can see certain regions that already show high KW and may continue to have regional increases in KW. So we're currently working on writing a manuscript and really diving into what are the regional differences in these two measures, where are we actually seeing group level differences between young and old, but I just wanted to share some preliminary results totally in the opposite direction. So I think even though we haven't necessarily obviously solved, the controversy, I think what we did do is actually perform both ASL methods in the same group. We had distinct younger and older groups. There's been a recent study that also did this in midlife to older groups, and they also saw a inverse relationship between the two measures, right? So now there's more studies like ours and like this published study that are showing these inverse relationships. And I think from a methodological perspective, it's really interesting because, trying to understand differences, for instance, in like T2 between younger and older. Maybe the tissue T2, there's also oxygenation differences, which affects T2 values between these two groups, and we have to account for that. Have we really rigorously harmonize the readout and the image processing steps, including smoothing? That hasn't been done in the literature yet. So both on the physical contrast mechanism, like T2, magnetic susceptibility, as well as image processing, I think there's a lot of harmonization to do. I'm not convinced yet that necessarily any of it will completely address this discrepancy. But I do think that no matter what, we can start understanding some of the other physiological correlations with each of these measures, and that together they might be able to give us insight in longitudinal follow-up. And especially if we can compare with DC MRI, I think the important thing perhaps is understanding some of the physiological correlates, trying to understand imaging differences, but in the end, they may not still be exactly concordant, and that's okay. I think that could be actually giving complementary physiological insight. And then, there's a whole slew of statistical approaches that we can try to understand, hey, which one actually contributes and in which regions would help predict cognitive decline in certain domains. I think that's a really rich area that I'm excited. Despite, maybe the confusing initial pilot results, I think it at least lays the foundation that we are able to do these methods. And in the last few minutes, I just want to touch on how, again, what Dean mentioned is like, I think ASL and in general, physiology is a new avenue that a lot of studies are looking into, and I'm really excited about it. So ADNI-4 had a long process through which it integrated single delay, pulse-label delay, and ultimately a few years ago proposed a multi-delay post-label, multi-post-label delay ASL approach in its harmonized protocol, which I think, again, gives more robust CBF measures than ATT. And what's exciting is also, the more recent ADNI cohorts are more diverse in terms of targeting like underrepresented groups. That's something our UC Davis ADRC is really committed to. For instance, our current active cohort is 26% Black and 28% Hispanic. And I think it touches on many of these studies as well that do adopt ASL. So we already talked a little bit about Clarity. I think Clarity has certain criteria by which, for instance, cognitively unimpaired enrollees need to fulfill criteria like to either have vascular risk or underrepresented in terms of like education, where they're from, race, ethnicity, etc. And diverse VCID is led by UC Davis by Charlie DeCarli, our center director. And it's really focusing on white matter hyperintensity lesions. And this is targeting 30, like a third Black, a third Hispanic, a third white, with the focus on lesion evolution. So I think there's a confluence of studies that are incorporating ASL and particularly ADNI-4 ASL protocol. And I'm really excited to see what it looks like in a diverse cohort. And those of you who went to AAIC will probably also remember that US Pointer was a really big study that received a lot of attention in terms of its results this year. I'm not involved in Pointer, but several of our core leaders at the UC Davis ADRC are co-investigators. And this was a 2-year lifestyle intervention, like cognitive, physical, and diet intervention funded by the Alzheimer's Association, and then with a almost 50 million ancillary imaging study funded by NIA. My colleague, Young-Kyu Jung in radiology here was part of the imaging study, and he's already started to incorporate, for instance, arterial transit time measures from ASL and understanding Framingham risk score in the Pointer study. So ASL has been part of the imaging component. But my final slide is just this really exciting result from Pointer at AAIC, which is that a healthy lifestyle can help people at risk for dementia. So as I mentioned, this was a 2-year randomized controlled clinical trial. I think the enrollees were cognitively unimpaired, but they had a relatively sedentary lifestyle upon entry. And participants were either assigned to a self-guided or structural lifestyle enhancement program. And I was really struck that the cognitive function improved over the 2 years for both intervention groups. Like when I looked at the results, and I'm not a cognitive expert, but I seem to remember many of the anti-amyloid immunotherapies, right? The way that they evaluated was slowing of cognitive decline. This was actually a cognitive improvement in both groups, which was phenomenal, and that the group receiving structured support, so more intervention like meetups, accountability with study partners, family, etc., showed the greatest benefit in protecting cognitive decline from normal decline in 2 years. So this is all to say, I think with these sort of interventions that I think anyone can have access to, and the fact that we have imaging underlying this, like in tandem, that is currently being analyzed as part of Pointer, I think it's very exciting for the future of brain health and healthy aging. And with that, I just want to encourage everyone to, continue exercise and eating well as much as you can and find a buddy to do it. And with that, I just want to thank your attention. These are some of our collaborators and a photo of my group. Thank you very much.
Awesome. Thank you so much, Dr. Fan. This was amazing. I know we asked some questions during the presentation, but I'll also open the floor. If anyone has questions right now, feel free to unmute or put them in the chat as we still have a few minutes.
Hi, this is Miriam from Neurology. Thank you so much. This is a wonderful presentation. I had a question regarding water exchange measurements, and if you could share some tips on this. So my question is about the water exchange map resolution, and if you have some advice on addressing the image resolution, sometimes that is not great, and especially when you want to do like ROI-based measurements in the analysis.
Yeah, I think that is a severe limitation of the versions that we have been working with. I think, for instance, the USC group, their diffusion-weighted ASL, I think the resolution might be, I think the thickness, for instance, is like 8 millimeters. I forget off the top of my head, but it's really challenging spatial resolution because, again, ASL itself is low signal, and then now you're trying to separate different compartments of the original low signal. So that means larger voxels and there's different amounts of smoothing applied by these different methods. So I agree with you, Miriam. This has not been studied extensively. I wouldn't be surprised if the sequence developers, especially at USC, for instance, they have access to 7 Tesla and they've been really thinking about ways to achieve high resolution, just standard ASL. So I wouldn't be surprised if with some sort of maybe machine learning approaches, advanced readout approaches, we might be able to improve it in the future. But I would completely agree with you that it is a severe limitation of the current method. And it's not, especially the smoothing aspect, is not harmonized between the 2 ASL approaches. So.
Thank you.
Hi, this is Ning again. I also have another technical question about the multi-echo T you're choosing. So you're choosing using a trust-based, like T2 preparation, or the multi-echo is accomplished by like multi-echo acquisition?
That is, I think it's a multi-echo acquisition, and I think there are 2 aspects of the acquisition. So there's one version, sorry, there are 2 acquisitions that go into the T2 ASL. So one is single echo, but many Hadamard post-label delays so that you can get reliable kinetic fitting and ATT measures. And then the other, I think it has fewer post-label delays, like 3 Hadamard, but it has multiple echo times. And I don't remember exactly how the T2 contrast was enabled. Like it might have been a prep, but T2 prep module, but I think in the end you get multiple echoes with a few post-label delays, and then you integrate it with the single echo with higher number of post-label delays. So it's a little more complicated, and another reason why the physical contrast mechanisms are perhaps challenging to compare across the 2 ASL.
Yeah, I'm asking because we use the latent trust sequence to do T2 prep, but I have difficulty to modeling that. So I was hoping that you may have more experience. I'm hoping you do the acquisition side of the T2 multi-echo. So hopefully that's because we are thinking maybe you want to switch on a multi-echo on the acquisition side instead of T2 prep. So I wonder if you have any like suggestion on that.
I don't have any immediate suggestions, but I think it's certainly a different context, right? Because the trust measure, you're trying to get a global sagittal sinus measurement. Is that right?
This one is not it's a global T2 prep, but it's not a sagittal sinus, but you can just thinking of the T2 prep. Yeah.
Yeah.
I'm still thinking maybe the CSF contamination because the voxel is so big and CSF has different T1, T2. And so I'm thinking maybe that's a contribution, and they have background suppression too. That make the things very complicated. So I'm wondering if multi-echo acquisition would be better choice than T2 prep.
Yeah, I completely appreciate your concerns, and I think that's very insightful. We are not the sequence developers, so I'm happy to, just brainstorm, and also from the modeling perspective, what we can do. But happy to connect after about it.
Yeah, thank you.
I apologize, I don't have an immediate answer.
Awesome. If anyone else has any questions in the last minute, I had one quick question. Just what you're talking about with ASL, being able to capture those subtle changes in cerebral perfusion, not using contrast agents. Do you think it has the potential to be a reliable early biomarker for dementia risk, like maybe even before people's clinical symptoms become apparent?
I think it could be a reliable early marker that you can track from an imaging perspective, but I also think it's not uniquely specific. So like someone who has vascular, well, let's just say hypoperfusion and ASL has been implicated in so many other, vascular risks, many neurodegenerative diseases. So I think it's not going to be itself diagnostic or anything like that.
Mhm.
I mean, you'll probably see regional perfusion patterns that, for instance, can distinguish aging versus AD.
Right.
It's going to be very heterogeneous, but whether it could be reliable enough to, for instance, monitor therapy or intervention effects, I think
Mhm.
that's definitely possible. Yeah, so I'm very excited.
Awesome.
Mhm.
Thank you so much for answering that.
Good question.
All right. Well, I think that brings us to a close. This was an amazing talk. Thank you so much for coming. We had a lot of participants, especially people coming, especially for an August talk. So I was happy to see that. But yeah, thank you so much, and we'll be in touch. And if anyone wants to connect, you feel free to reach out to me, and I can connect you guys. And I hope everyone has a great rest of their day.
Thank you.
Thank you, Dr. Fan.
Awesome.
Cheers.
Thank you so much.